All right, why don't we get going so we can listen to these unbelievable speakers. This is going to be a great session. So welcome to our 11 o'clock presentation panel discussion on preventive care and health equity. This is where Wisconsin truly shines, and I look forward to hearing from Amy Kine and Honey Reddy, representing our two outstanding Wisconsin research institutions, the University of Wisconsin-Madison School of Medicine and Public Health, and the Medical College of Wisconsin. Since our sponsor is also moderating, I'm going to have all the speakers now come out, and then Lisa Cadmus-Bertram of All of Us Research Program will first be saying a few words as a sponsor before starting our panel discussion. So please welcome our panelists.
All right, here we go. Good morning. I'm happy to be with you here this morning. My name is Lisa Cadmus-Bertram. I'm an associate professor in the Department of Kinesiology at the University of Wisconsin-Madison, and I'm here representing the team from All of Us. This year's summit theme, The Next Generation of Personalized Medicine, is aiming to address the idea that healthcare should not be a one-size-fits-all solution. We imagine a future where prevention, treatment, and care are tailored to you. We believe that future starts with research that includes all of us, and that's where the All of Us program comes in, underway at the University of Wisconsin-Madison and UW Health. The All of Us research program is a historic effort to gather data from 1 million or more people in the United States to accelerate research and medical breakthroughs. As a program focused on precision medicine, we are working towards making healthcare and health research more personalized with considerations for an individual's environment, lifestyle, and family history. To do this, we have a three-tiered approach that you see here that includes nurturing relationships with our participants and their communities, delivering the largest and richest biomedical data sets, and building an ecosystem of communities, researchers, and funders. Already, thousands of Wisconsinites have joined the All of Us research program. With over 27,860 people that have joined this community, we believe that the next health discovery could come from Wisconsin. Across the United States, more than 717,000 people have already enrolled in the program. Of these 717,000 individuals, more than 511,000 biosamples have been collected and 399,000 electronic health records shared. Because of the dedication of All of Us to revolutionize healthcare, there have been over 200 scientific publications and over 7,300 active research projects using the All of Us data. And while we have these amazing successes due to this effort, it's truly just the beginning. We need people from all backgrounds, neighborhoods, experiences, health conditions, or no health conditions to join All of Us. And this is where you come in. First, feel free to enroll in our program yourself, but we believe that this program is a partnership with participants, and that's what we want to do, to be able to partner with wonderful companies that are here today. We'd like your organizations to join this exciting program by becoming an All of Us research program partner. Together, we can explore ways to bring all of us to your company's community. This could be through lunch and learns hosted by All of Us, distribution of materials, or enrollment events that come to you. We want to help empower you and your staff to take charge and play a part in revolutionizing healthcare throughout Wisconsin and across the country. A unique aspect to this program is that participants can directly learn more about their health. The program can provide participants with DNA results over time for those who want to see them. This includes information about genetic ancestry and traits, whether a participant may have a greater risk of developing certain hereditary diseases or health conditions, and how your body might react to certain medications. Kayla Lemon and Yvette Craig are with the All of Us site, and they are here today. Kayla and Yvette, are you here? Yep. All right. Thank you. So feel free to find either Kayla or Yvette and learn more about the program. They will be at the All of Us table, and I hope that you'll go speak with them. They'd love to talk with you and help you understand more about how you or your organization can become part of the program. So thank you so much for providing me the opportunity to speak with you about All of Us. On this screen are our two principal investigators, Dr. Beth Burnside and Dr. Dorothy Farrar-Edwards, who also want to thank you. You'll see our QR code there that you can use that will bring up our website, or you can just speak with Kayla and Yvette. With that, I'd like to introduce our first panelist for today. Dr. Honey Reddy is a professor in the Department of Pathology and at the Clinical and Translational Science Institute at the Medical College of Wisconsin, as well as their Division Director of Precision Medicine and Cytogenetics. Welcome, Dr. Reddy.
Thank you, Lisa. Morning, everyone. I'd like to thank BioForward for the opportunity. It's an exciting times that we are in. As Lisa talked about the All of Us program, clearly the drive in the field is to be able to get a significant amount of genetic information, genomic information about individuals, and as Amy will talk about, how do we connect it together, right? So how do we move from precision medicine, where we are looking at specifically targeting changes at the DNA level, to how do you view it from a perspective of an individual in personalized medicine, taking into consideration their lifestyle, the environment, and so many other social factors. So I'll be talking today about polygenic risk scores. I had the privilege of working with a very specific group of individuals on behalf of the American College for Medical Genetics and Genomics to be able to put together lab as well as clinical considerations for the implementation of polygenic risk scores in the population. So I just wanted to start a little bit as we talked about precision versus personalized medicine. So as I said, precision medicine is where you're able to do treatment and management of disease based on molecular diagnostics, right? So that's my bread and butter. I oversee a clinical lab that does genetic testing both for germline disorders as well as acquired cancers, and being able to identify a therapy based on our findings within a molecular diagnostic lab. So you can use multiple technologies like listed here, right? There's genomics, there's transcriptomics, so looking at the DNA level, looking at the RNA level, trying to identify those different variants. But when you put this in the context of personalized medicine, you want to be able to take the data that you're getting from the different technologies, so epigenomics, proteomics, metabolomics, et cetera, but then putting it in the context of the lifestyle and environmental exposure an individual has to be able to provide that holistic picture for treatment and management. And so if you take a look at the current paradigm from an individual perspective, to be able to expand this to the population, keeping a holistic view of those different factors that needed to be taken into consideration. Because when you're looking at genetics, lifestyle, and environment, you're able to do predictions of risk based on behavior and exposure to ultimately address what is required to maintain health for a given individual or a population. So for those students who are not very familiar with genetics, this is me as a teacher. It's like genetics 101. So pardon me if it's too basic, but there's those variants that you can inherit from your parents that can be passed on to the next generation. It's present in every cell. It's also present in the gametes, so obviously that's why you're able to pass it on. And then there are those acquired variants that an individual acquires within their own lifestyle that's very specific to specific organs or specific cells and tissues, and that's where you have for example, the different cancers. And so a few examples on how these genotypic changes can impact your phenotype, right? So you could have neutral changes like eye color, birthmark, that do not impact an individual whatsoever in a negative manner. You could have harmful variants, that could end up with cancer or constitutional disorders like cystic fibrosis, epilepsy, etc. Or you could have beneficial variants where you have the variant for sickle cell anemia that actually gives you resistance to something like malaria. So it's a perspective of understanding what those variants are and how they would impact the phenotype. So it's not necessarily always only one gene. So you have your classic monogenic disorders, like cystic fibrosis I mentioned, sickle cell anemia, et cetera. But you could also have multiple genes that are contributing to the same phenotype, as you would see with celiopathies, epilepsy, autism, et cetera. So autism is between the oligogenic and the polygenic. Polygenic, on the other hand, is significantly influenced by the environmental factors that one is exposed to. And so when you put that in the context, you're actually able to see moving from your monogenic disorders, which are primarily diagnosed through linkage analysis. You could have the oligogenic and the polygenic that can be informed based on genome-wide association studies, right? That are also having to be reviewed in the context of the environmental exposure. So how do you move from genetic variation to precision or personalized medicine? So on the left, you have your monogenic disorders, like I talked about, single gene, single disorder. So for the biochemical students out there, right, it was always one gene, one enzyme hypothesis. That's how I remember in school being taught. But when you look at genetic causation, it's clearly, unless it's that classic single gene phenotype, more or less what we are seeing now is an oligogenic or a polygenic phenotype, right? So when you have your monogenic disorders, you do your genetics, and then that then tells you exactly how to do treatment, and mostly it's management of disease. But when you come to your polygenic, you have to put this in the context, like I said, of the environmental factors, the behavior, the social environment, and things like that, to be able to identify what would be necessary for treatment and management of disease, but also to be able to call out preventative measures. And that's where polygenic scores come into play, right? So this is one of the publications. I'll talk about the next one as well. So 2 different publications that came out in March of this year that was driven by the American College for Medical Genetics and Genomics, and the one was on the clinical application of polygenic risk scores, and the other was the lab implementation. So I just wanted to bring across a few salient features of each of these documents. They are freely available. You don't need any kind of membership. ACMG puts all its recommendations and guideline statements open access. So if you all want to get more information on this, it is available out there. So when you take a look at clinical implementation of PRS, there are 8 different points that were put across. One, that PRS test results do not provide a diagnosis, and that's the key. If you remember when we talked about monogenic, we said you identify a variant, and that is possibly what is considered to be the genetic causation for disease. But in polygenic, you're not doing a diagnosis here, right? What you're doing is providing a statistical prediction of increased clinical risk. Having a low PRS score based on the computational algorithms does not rule out significant risk for the disease. And this is where one has to think about the behavioral norms, the social considerations, the exposure, environmental components, etc. If the risk prediction is derived from a population that is different from the patient being tested, one has to be very, very clear that it has a poor predictive value. And so, for example, if the algorithm is based on a purely African-American population, it will not work for someone like me who is of Asian descent. So just something to keep in mind, that the way the algorithms are being built has to be customized to a particular ethnicity for it to make sense and for it to be close to accurate, because one has to remember these are risk predictions, so they are not truly accurate like when you're doing diagnostics with molecular testing for monogenic disorders. Isolated PRS testing is not the appropriate test for clinical scenarios in which a monogenic etiology is known to be present or suspected. And an example would be if you have a baby present with all the signs of cystic fibrosis, you should be doing testing for the CFTR or the cystic fibrosis gene. You should not be doing a polygenic risk score. So this is where I think it's a understanding and a collaboration between not just the testing lab that gets the order and says, oh yeah, we can do this. It's a collaboration between the clinical geneticists, the ordering providers, the patients, the labs, the genetic counselors. So everyone together has to understand what's the best way to do this testing. Before testing, one should discuss the outcomes of these tests with the patients themselves so that they have a clear picture that this is not diagnostic and how it can be potentially used for the future. PRS-based medical management should be evidence-based, right, in the sense that please remember that these algorithms have to be built with certain considerations put in place. So it can't just be built out of the blue. And then clinical follow-up with the PRS results should be consistent with the best practices outlined. So PRS is usually, as they are looking at in the field, being considered for increased risk for diabetes, increased risk for cardiomyopathies, increased risk for cancer. So when you look at it, it has to be done in the context of a clinical environment, not in isolation. So direct-to-consumer testing is not recommended with polygenic risk score testing. And then I think one of the key features key points that we really discussed significantly was the use of polygenic risk scores for pre-implantation genetic diagnosis. So when you go looking at the field of IVF, infertility, things like that, where pre-implantation genetic diagnosis now starts looking at Down syndrome, presence of Down syndrome, or the risk for any of the recessive or dominant monogenic disorders. We do believe very strongly that PRS is not ready for that. And there is an independent document that was put out by the ELSI, or the Ethical, Legal, Social Implications Committee of ACMG, on this particular topic, that PRS is not ready for PGD right now. The second publication we had out was the lab perspectives. So there is, when you take a look at genetic testing, there is folks in the lab who develop a test, put it out there, put out recommendations, but it is the clinician or the ordering provider who takes those results and determines what needs to be done with treatment and management based on those results, right? But it's also important for the lab folks to understand how is it that they are developing these tests. They have to have not just an analytical validity that yes, the quality is spot on in terms of the sensitivity and specificity, but what they also have to understand is the clinical utility of any test that you're developing. Will it make sense to offer this test to a specific population or an individual? So the analytical utility, validation, and the clinical utility are very important to be considered from a lab perspective. So again, just to belabor the point, right, when you're looking at it from a lab perspective, PRS is an estimate of an individual's susceptibility to develop a disorder, and that should be kept in mind. It is not diagnosis. It is not giving you genetic causation for disease, because it's based on genome-wide association studies. And so this particular document looked at the general considerations, the considerations for implementing these tests, the appropriate criteria for the reporting of PRS, as well as the scope and limitations of PRS testing. And so from a measurement perspective, that if you're bringing up this assay, what do you need to look at, and what are the key points? So we say that at this point, there is no PRS currently available from a clinical lab that has been shown to deliver equally informative and accurate results across different ancestries. And that's the key, is that are they looking or taking into consideration the different ethnicities or the different genetic ancestries as they are building? And we are of the standing that no, there is no current PRS that does so. The extent to which the differences are clinically and ethically meaningful should be done in collaboration with community partnerships. And even for monogenic disorders, there are always these very close partnerships, for specific ethnic populations with their clinical providers or the genetic society. So we recommend that similar partnerships are built for polygenic risk scores. And therefore, given the potential of risk, there is a chance that it could actually exacerbate the health disparities, because if it's not developed correctly, you can have inequality across the different populations. From a clinical utility perspective, we do recognize that there are no large-scale studies that actually establish the clinical utility of PRS. And again, we belabor the point that it should be disease and population specific. From a reporting perspective, we do say that integration of polygenic risk scores into clinical reporting, they've implemented in different settings, clearly not the best practice, but if they do include reporting, then it has to be done in a proper way, and it has to be consistent across different labs. So if an individual takes a report from one lab, goes to another provider, moves to a different state, they are still able to understand the context of those results. So it has to be commutable across practices. And then limitations as well should be outlined in a report, because this is such a new field that the patients as well as the ordering providers need to understand the limitations associated with reporting of polygenic risk scores. Some other considerations, there needs to be greater diversity in genetic studies, and this is, again, the same point. It has to take into mind the different genetic ancestries. And it's also, these are DNA-based tests, and so considerations between genetics, genomics, and epigenetics has to be taken into consideration. You're not looking at methylation when you're doing polygenic risk scores. You're not looking at acetylation of the DNA. You're not looking at changes at the RNA level, so one needs to put all of this in context as you're developing it. And then back to the pre-implantation genetic diagnostics, that we are not ready yet. And there has to be a lot more information gathered to be able to translate this to that particular aspect of genetic testing. And so we come back to this point where, I'm just trying to put it all together, that the point to take home is that it is indeed a collaboration. It's not just the lab that's doing the testing. It's not just the ordering provider. It has to include education of the patient, education of the provider, education for the labs, and education for the patients themselves. So understanding what goes into this, the involvement of the community to make sure that those variations in genetic ancestries is addressed. Significant amount of research, like Lisa talked about, the All of Us research program. So dedicated efforts towards that. And then from a clinical perspective, the labs and the providers working together. So at the Medical College of Wisconsin, I think we are looking to address all of this. So there is research very specifically looking at precision medicine. We do have the All of Us program there as well. But from an education perspective, we have the Masters in Precision Health, precision medicine, that we have lab genetics and genomics involved. We look at cancer precision medicine in different aspects. So that's what I'm talking about, that educating the next generation on the required skill set to make sure they are doing it right, I think is very critical. And I'd like to end by acknowledging all the working group members. It took us 2 years to get this put together, and it was very, very satisfying to be able to get a document. So just to let you know how these documents get published, we have to put in a proposal, which I did in 2020. It took me a year to get board approval, because they wanted ethics, they wanted clinical practice people, they wanted genetic counselors. So we got our group together, and then we decided to split it into two. Once the document is written, it actually goes through Lab QA committee for reviewer. That's 24 members. Then it goes through the practice committee, which is another 10 members. Then it goes to the board. From the board, it actually is put out for 30 business days for all members, which is, I think, close to 1,000, 1,500 members to comment on that document. And after that, we address all those requests, and then it gets published. So it is a huge task getting these documents out, but it is extremely satisfying, and we look at it as our contribution to the field to be able to make sure the thought process for genetic testing is in place. So I thank you very much again for your attention and the opportunity.
Thank you, Dr. Reddy. That was a wonderful discussion of the difference between precision and personalized medicine, as well as polygenic risk scoring. I'm happy to introduce our next speaker. Dr. Amy Kind is an Associate Dean of Social Health Sciences and Programs at the University of Wisconsin Madison School of Medicine and Public Health. Welcome, Amy.
Thank you. Thank you so much, Dr. Bertram. And Dr. Reddy, you couldn't have set the stage better for coming into this next portion of the talk. So I'm very grateful to have the opportunity to chat with you today. Thank you to the organizers. Thank you to the entire BioForward leadership team for inviting me and for putting this very important session forward. I do think in almost every conversation we have, health equity should have a place at the table. And so I'm grateful that this session was pulled together. So today we are here to talk about the exposome and how the exposome forwards us into perhaps what I like to think of as a next generation of precision medicine. So when we think about precision medicine, you heard Dr. Reddy give a wonderful presentation about some of the basics of genomics. Another way to think about it from a precision medicine standpoint is traditionally it involves deep phenotyping of an individual. What makes that individual's biology unique, the genomics, other omics, and yet health, we know, is a combination of genes plus environment. In fact, there increasingly is suggestion that the environment may outpace the genes in terms of its contribution to health outcomes for a whole host of complicated diseases, especially the polygenic diseases that Dr. Reddy mentioned. So because of this, environmental exposures need to be thought about, they need to be quantified, and most importantly, they need to be addressed. It's exciting because environmental exposures are technically modifiable. These are points of action we can take, but there are also items that we can measure. And could it be that the water that you drank and the air that you breathed when you were 7 is setting you up for the breast cancer that you might have when you're in your 40s or 50s? And should we be thinking about this when we're prescribing diagnostic treatment tempos or other types of therapies? But first, we need to think about the construct and start measuring the construct. This whole field is considered to be the exposome. This is a term that was just coined not even about 5 years ago. And it is the totality of factors that are external to the biological individual. It includes factors as diverse as the microbiome, all the way through to structural and societal inequities. As you can see here, it can be thought of in 4 components: ecosystems, social factors, physical and chemical exposures, as well as lifestyle. At the University of Wisconsin School of Medicine and Public Health, we've established the Center for Health Disparities Research in order to study the exposome, in order to build some of the special technical expertise that's required to link these social factors, quantify these social factors, and link them to fundamental biology. When we think about examining the exposome, it can be simplified into 3 fundamental steps. The first is taking social factors across the life course or other types of external exposures and quantifying them in a rigorous and reproducible manner. Many of these types of things are traditionally studied through survey. This can be one method of quantification, but we have others. And we have other tools that I'll talk to you about today that revolve more around geospatial analytics and precision geography than survey methodology. Next, we need methods by which to link these exposures to biology. So how can we take some of the very fundamentally and exquisitely phenotyped tissues and biorepositories that are currently in existence and add back social factors? So often these biorepositories are de-identified. How can we add back some of these exposomal factors in order to make meaning of the findings that we have? And then fundamentally linking these research constructs to action, both from a pharmacologic and therapeutic standpoint, but also from a social action standpoint. So first of all, quantifying exposures. I don't brag that much, but I have to, I'm a Midwest girl through and through, but I'm pleased to say that we truly are, Cheddar truly is the national leader in social exposome quantification and study. We provide metrics for the full federal United States for its health equity benchmarking and cohort studies across the National Institutes of Health as well as across the US use our metrics in order to add meaning and add social exposome quantification. Cheddar stands for the Center for Health Disparities Research. It's a cheese name, which is appropriate for the University of Wisconsin. But we are the creators and curators of a tool called the Neighborhood Atlas. And through the Neighborhood Atlas, anyone can access a metric called the Area Deprivation Index, or ADI. We construct this metric. It includes 17 different measures of the social determinants across very precise geospatial metrics, or geospatial areas. It's a rank-based score that's time concordant. We can construct these all the way back to the early 1900s, and it is incredibly strongly linked to health. We distribute this metric, and right now it is the most commonly used disparities-aligned US social exposome metric in our country. There have been over 1,200 peer-reviewed studies just since 2019, and when I looked at Google Scholar just yesterday, we had over 850 publications just in the last about 12 months. So most of these are not through our team, they're through other teams that are using these metrics and learning about these metrics. On our tool, we've had over 1,000 downloads each week of the full data set, and these are 79 million data points that are available to you if you are interested. And as I mentioned, it's been used extensively in state and federal policy, which I'll share a little bit more about in a moment. We also provide customized mapping in that any individual can interact with these metrics, even if they don't have a data science background. If you can use a smartphone mapping app, you can interact with these metrics and learn a little bit about the areas that you're interested in. This happens to be the county of Milwaukee, the entire county. Typically, when we think of exposomal metrics, often you see county rankings or county level scores, but a county is a huge, huge, huge geographic area with a whole host of different types of exposures. When we use a much more precise metric like the ADI, you can see a variation of pattern across the county. In this case, the red areas are the most disadvantaged or highest ADI, and the blue are the least. Those red areas are clustered within inner city Milwaukee, and they map onto areas of historic redlining within that city, and they also echo many of the areas of adverse health outcomes and disproportionately impacted populations. This is the state of Wisconsin. Note that not only are inner cities impacted by these high ADI scores, but our rural communities are as well. Our native communities, our rural communities, and these patterns are ones that we see across the United States. The true power of these metrics, though, comes when we can link them to biology. And as I mentioned, this can be a tricky thing to accomplish, particularly when we're dealing with biorepositories that have linkages that have been broken. This technique is called social biological phenotyping, and it really allows us to expand existing programs or existing resources in completely new ways by adding on social factors and understanding more, particularly about exposomal exposure and some of the associations that can go with that construct. So just one example, we have many, many examples in the brain health space, but I'll give you just one. And this was some work that was led by a scientist of mine who's now an assistant professor at the University of Wisconsin, Dr. Ryan Powell. So we partnered with the University of California, San Diego Brain Bank, as well as our University of Wisconsin Brain Bank. And with these brain banks, typically individuals, when they die, they have donated their brains to science with the hope that future generations will find cures to some of our most devastating neurodegenerative diseases like Alzheimer's disease. So this is truly quite the altruistic gift, and we want to make the most of this wonderful gift that these participants have given. Yet these tissues tend to be very, very deeply characterized from a biological standpoint. Most of them have been completely genotyped, and a whole host of other assays have been run. Yet no social factors are often available. In fact, with these two brain banks, and UCSD is one of the oldest in the country, there was no social factor characterization at all for these tissues. We don't understand their education, we didn't understand where they were living, we didn't even sometimes understand their racial and ethnic background. So we wanted to find out, to test the feasibility of whether we could add back some of these social characterized factors. And so what we did was get the autopsy reports that were collected at the time of brain donation. These individuals in this particular cohort could have donated as early as 1993. We didn't go earlier than 1993 because nothing was in an electronic format prior to that. Everything was in kind of yellow legal paper, and we didn't want to quite go back that far. So we started at 1993, and we went through 2016, got the autopsy reports, and we took the address at the point of death from those autopsy reports. We used a historical method to geocode in a time-concordant manner those addresses, and then we could link them to time-concordant area deprivation indices, or ADIs, that our team can build. And what we found in a dose-dependent manner is those individuals who were living at the time of death in the most disadvantaged neighborhoods by ADI had a markedly increased burden of amyloid plaques and tangles, along with the Alzheimer's disease, these hallmarks, these gold standards of Alzheimer's disease. And we could see this going up in a dose-dependent fashion. This study was not designed for that finding. We were just designed for feasibility, and yet the signal was so strong it came through. Since that time, this finding has been replicated in a number of other places. And note the publication date on this. This just came out in JAMA in 2020. And so this field is moving forward so quickly, this addition of social factors and trying to understand more about how they're linked to fundamental biology is a new frontier. And yet this is just cross-sectional. And what we talked about with the exposome was a longitudinal vantage point. Where did we live across our life course? Could we add life course data back to individuals who are already dead? Is that possible? And yes, it is. This resulted in the neighborhood study, which is one of the largest studies ever funded by the National Institutes on Aging in the social exposome space. This is a $37 million R01 that we were able to compete for. We are the coordinating center at the University of Wisconsin, and we have 23 sites across the United States. What we are doing is linking the social exposome to this biologically phenotyped tissue in order to better predict risk of disease. And we think about this through residential history construction. So if you look at the cartoon on the bottom, when you think about where you were born as compared to where you die, you may go through a number of different residences across that time. Well, what if you had adverse exposures in the beginning of your lifetime as compared to the middle of your life course or the end of your life course? We need to be able to build these geospatial frameworks across an entire lifetime in order to then use those frameworks to link on different types of exposures. In this case, we're linking time-concordant ADI, but we could just as easily use these foundations for linking in data on pollution, data on school quality, different types of adverse exposures through environmental catastrophe, and the list goes on and on. We do this through public data tracing, which I'll talk about more in just a moment. These are our different sites. You can see we're all across the United States. Our site within Fort Worth and Dallas, Texas, will be one of the largest cohorts of Mexican Americans and immigrants within this brain health space. So we have good diversity across a number of domains and good geographic diversity as well. We are just in the second year of this study, but we've hit the ground running. Some of the things that make this study particularly unique is we have developed data use agreements with each of these 23 sites to share protected health information. So we can bring in names, addresses, dates of birth, dates of death for all of these individuals, and that was a massive lift. We have tailored data use agreements for each state in this case in order to allow for that data sharing and have built customized cybersecurity platforms in order to secure the data and process the data in a very safe manner. We talked about the challenges that HIPAA poses to data sharing and to some of these data, these exposomal especially types of assessments, and yet we have been able to launch this and move this forward. At this date, we have over 10,000 brain bank decedents data that have been transferred to us at the University of Wisconsin. Each of these individuals is going through a full public data tracing in which I employ a team of archivists that use traditional historical methods, how about that, to trace these individuals back from the point of death all the way to the point of birth. And with that, we find birth records, we find marriage certificates, we find military records. We have some scientists that are doing additional assessments looking at military exposome and adverse exposures in Korea, Vietnam, and World War II that could be linking to different types of neurodegeneration as well. But, so it's very exciting, but it wouldn't be possible without this extensive PHI cybersecurity data platform. And as these data come in, our vision is to not only link these data, store them securely, and link them through these precise geoanalytics to exposure, but then share back de-identified data to other resources so that other groups don't have to reinvent the wheel. We are trying to build bridges and not silos. I'm happy to answer more questions about this, but this has been of great, great interest to the NIH, and to our knowledge, we're the first group in the country doing this. But also linking research to action is important. So the NIH across multiple institutes is using the metric that we provide through the Neighborhood Atlas, the ADI. We have a number of industry partnerships that are born and growing. Professional medical societies are using this metric to measure and quantify health equity. Most recently, the Society of Thoracic Surgeons, looking at their performance for patient capture and outcome. We've worked with a number of private insurers, including Michigan Blue Cross Blue Shield, and a number of other plans across the United States as they think about quantifying equity. State Medicaid programs, and then the US Centers for Medicare and Medicaid Services have announced that the ADI is their official health equity benchmark for their ACO REACH program and a number of other programs in order to be able to more efficiently target resources to areas of need and to be able to measure these aspects of social exposome in a number of different ways. Finally, just one local program. There's hundreds and hundreds of programs across the United States that are using our metric, but I just wanted to highlight one local program that might be of interest to you. The city of Milwaukee, just in May 1st, 2023, you can see here, announced their new waterworks prioritization plan, which uses our metric. They were able to download the metric from the Atlas, and they built this. I was not involved with this, but what they are doing is targeting neighborhoods with the highest ADI scores in order to prioritize which roads to dig up and which pipes to change. So you can see the old lead pipes here, the new nice copper lines going in, and they were able to do this geographically. This allows for more efficiency as they move forward. They can target neighborhood by neighborhood at a time, and they have a mathematical model by which to make these decisions in a way that has been helpful to them. I just did a visit to Milwaukee as part of my Associate's Dean's role, and we were hearing from some of the individuals who are involved with these lead exposure groups that this type of philosophy of targeting within the city of Milwaukee, at least, is expected to be expanded to other types of programs. Additionally, hot off the presses, this just came about a week and a half ago. This website was just launched. I only know about this because I was asked to be on the National Advisory Board for this new office. So the National Institutes of Neurological Disorders and Stroke just launched their Office of Neural Exposome and Toxicology Research. They are looking to do a major initiative within the exposome and brain health space, and they are focused on all 4 of those domains that I showed you in the social exposome cartoon at the beginning. So thinking about environment, thinking about social factors, thinking about behavior, and thinking about toxins. I have been brought in because our team is really the expert, the national expert in the social exposome space, to help them think about those quantifications and think about the different types of structures and foundational policies that are required in order to propagate and to harmonize that type of research across the country. So I'll end there. Certainly the work that we do wouldn't be possible without a wonderful, wonderful team. I have to thank our over 70 neighborhood study collaborators across the country, the National Institutes on Aging leadership and staff that make this project possible. Of course, the participants that have given so much of themselves, and our Neighborhood Study Executive Committee, and then certainly our entire team at the Center for Health Disparities Research. We are a young center, but we are growing extremely rapidly. This is a hot, hot, hot field. It's moving very, very quickly, and certainly all of the work I shared with you today wouldn't be possible without our entire staff team and scientist team contributing. So thank you very much, and I'll end there.
Thank you very much, Dr. Kind. So I'll open up with an initial question and then hand it over to the audience and take your questions as well. I know that many people say that personalized medicine is really going to have a significant impact on clinical medicine by the end of this decade. And I'm curious to hear your thoughts. It's kind of a two-part question on what do you think are the biggest strengths or factors that are pushing us towards reaching that benchmark, and what are some of the biggest challenges that would need to be overcome in order for that to occur?
Okay, all right. So I think definitely we have to be there sooner rather than later. What I think, and again, my just personal views from being in the field, is collaboration. I think that is the key. Understanding that there is significant diversity in the population. I think where we struggle from a clinical genomics perspective, and I was talking to Lisa and Amy a little bit ago, was when we put out variant information from a molecular diagnostic test on a report and we say, this is a pathogenic variant, this is an actionable variant, this is a variant of uncertain significance, or this is a polymorphism, we are going by the information in databases that are biased with genetic and genomic information from specific populations, because the minority populations are not being included yet. But I think the positivity, or what we have to look forward to, is where programs like All of Us, or even smaller communities that are trying to do genomic profiling for specific ethnic groups to be able to then include this information in these databases. So collaboration and the coming together to understand that you have to be understanding of the diversity in the population, and when you put information out there, how is that being reviewed? I know that NHS in the UK has a program that's doing whole genome sequencing for their population. The Middle Eastern countries, Qatar, Doha, Kuwait, Saudi, are all also driving significantly huge programs to do whole genome sequencing of their population. So it's great efforts. The question is, will they then collaborate on a global level to allow us to have access to that data, right? That's the key. Everyone can do their own initiatives, but how do we come together to bring that information? Amy talked about PHI and HIPAA and all of those restrictions and constraints, but I think that with breaking down the silos, only then can we, it is no longer one country, one specific population, right? There is so much of diversity across the globe, and people just moving everywhere. So being cognizant of that fact, I think, is critical to being able to be successful with personalized medicine.
Yeah, I couldn't agree more. Certainly, I think our research resources need to reflect our population as a whole. And they have not done a good job of that thus far. I think all of us is taking some important steps. I think we do have a long way to go. I think in addition, with precision medicine, when I look at the burden of adverse health impacts across our society, often the populations that experience the greatest burden are not necessarily the ones that are benefiting from the treatments that are coming forward. So I do think that there's an opportunity, both from an industry standpoint, as well as an academic standpoint, as well as a societal standpoint, to think about those populations and to target those populations in a way that improves health. And these are tough questions. And fundamentally, it gets to the root causes of why health is different across populations, or when we think about these differences, another way to label them is as a disparity. So why do disparities happen? How do they come about? But more importantly, how can they be mitigated? So fundamentally, I'm an Alzheimer's doctor, I'm a brain health doctor, and we deal with a lot of neurodegenerative disorders that have no cures. We have some therapies that can slow things. There's some newer therapies that have come forward, and yet there's so much more to go. And we still need to understand the fundamental mechanisms that underlie these disproportionate burdens of disease. So why does a group, for instance, have much higher rates of Alzheimer's disease? What is that about, and can we change that? Can we change that trajectory, or are there different pharmacotherapeutic pathways that could be leveraged that could result in completely new cures, completely new pathways for discovery? And I would say in order to open those doors for discovery, we really need to think about these problems differently and build the bridges that Dr. Reddy talked about. And this is one of the reasons that the exposome is so exciting to me, because we can often measure factors that individuals aren't even aware of that they've been exposed to. And we can build these metrics backwards in time. Imagine going into a cancer clinic, and not only are they taking your regular history, but you are doing some sort of a residential history assessment in which you understand, wow, there were different types of exposure in my environment when I was young or in my midlife, and this needs to change the way I go about my care and my patterns of therapy. But I do think that that is the pie in the sky vision. You know, it's going to be a while before we get there. I think some of the advanced analytics hold a lot of potential in this space, but ultimately we have to think beyond our silos. We have to build these bridges and move together in collaboration.
Thank you. Let's have a question from the audience. Yes, in the front row.
I wanted to ask, are there initiatives to showcase any of this to the general population? Knowing that when I go to the doctor, if they don't ask me about, and maybe it's not there yet, but ask me about my residential history, ask me if I want to be genotyped or phenotyped, how do you know that you're not missing out on that?
I think that's a great question. I would have to say the exposome field is in its infancy, so we're not quite there yet. We don't even have the, our team is developing the analytic tools to deal with these complex data. Even historical geocoding is not something that you can just buy a program off the shelf. Like, we have to custom program all that work. But that's our vision for the future. We would love for someone to be able to step in and it just happens automatically. You know, some of these tracings theoretically can be done without even a person filling out a survey as part of their assessment with their consent, and if that's something that they want to do as part of their cancer treatment, for instance. So the potential is there. The technology isn't quite there, but we're working very hard to develop that. I'll turn it over to Honey, though, about the you get the genetics question.
So I think we'll definitely get there. I'm not sure if, you know, when you'll go into your PCP offices now, you'll actually have to fill out a survey on mental health. This was not something that was done 5 years ago, right? So clearly, the thought process, the understanding that it's not just the physical health, we also have to start looking at mental health, identify individuals who are in situations that need help. So clearly, like Dr. Kine said, if you're looking at where the exposome is right now, where genetics and genomics is right now, they are at a point where they are asked about or talked about only if needed, right? So with the way the field is moving, I'm sure in 3 to 5 years from now, because there are a lot of individuals, believe it or not, who just go and get their genome sequenced and then come into the clinic and say, okay, tell me what's wrong with me. And then the clinician will then come out to me, send me a note and say, hey, honey, we have this data, what do we do with it? I'm like, nothing I can do with it because you didn't sequence it in my lab. So I am not going to take the liability of evaluating it, right? So I think the understanding in the population that just getting your genome sequenced is not going to get you anywhere. But doing it the right way, maybe having conversations with your physicians. So I'm thinking maybe 3 years is too much to think about, but I think it's there. I think they are thinking about different aspects of a holistic well-being. So 3 to 5 years, I think we will be filling out those surveys on did you have genetic. So look at the 1 million people who are as part of all of us, right? They are having their genome being sequenced. So they will be able to fill out that survey and say, yes, we did have our genome sequenced. The concern there, and this is just from being in the practice, is in our lab, for example, if they come back and say, hey, can we reanalyze the data that we did for this kiddo from 5 years ago to see if there's something new? I say no. Anything older than 2 years, I don't think it would make any sense because the technology has changed so much in the last 2 years that I would think that the best way to address a genetic causation or understand for this kiddo would be to get a brand new sequencing done. You know, so the field is definitely changing at a very, very fast pace.
The question here, I think there was an article in the paper a couple weeks ago about, say, a newborn has some kind of malady that's been developing, and then there's a school of thought, well, should we do a full genome sequence on this individual to find the problem, or maybe look at the particular symptoms or signs that the person was expressing, and then do a focused look at it, knowing that, you know, you're going to get results quicker from the focused one than you would from the whole genome sequence. But then in the same amount of time, you may go off on a red herring with the focused one, and then not discover really what causes it.
So the simple answer to that is a lot of this is insurance reimbursement driven, right? So when you have a very clear-cut phenotype, then you say, okay, let's just run a dedicated panel and see if we can find the answers. But within our lab, as we do genetic testing, we see kiddos who've gone through a panel, who've gone through an exome, and then they end up doing whole genome sequencing. But now I think the way the field is moving is, originally it used to be microarray, which is a technology that looks at changes at the chromosome level. If you have a significantly impacted phenotype, you know, there are facial changes, they can't walk, they can't move, things like that, we would look at the chromosome level. But now the field is moving towards just doing whole genome sequencing. So there are programs out of Harvard, which is BabySeq, where they are, you know, sequencing a significant number of babies in the NICU. Rady Children's has a very good program that's looking at doing whole genome sequencing. We've actually established processes where we can do whole genome sequencing and give out a preliminary report in 7 days. So that's a significant progress. So our lab is, a lot of labs actually out there are able to do a preliminary report in 7 days. If we find that answer, we talk to the clinicians and we say this could be the reason. And then we take another week to finalize all our findings and present it. So the field is definitely moving towards doing whole genome sequencing. The NHS also has all babies being born, we are doing whole genome sequencing, just so we have that project. So newborn screening, right now they want to be able to do with whole genome sequencing, because currently the newborn screening is a heel prick, where you screen for anywhere between 23 to 50 metabolic disorders. Once you find something that's not right, then you go back and do molecular testing to confirm that that metabolic change is related to this. But, you know, like I said, the NHS is just doing all babies being born, let's sequence them. The BabySeq project in Boston, Massachusetts. So different examples. So we are moving towards WGS, and then from a reimbursement perspective, we actually now have dedicated CPT codes for whole genome sequencing, whole exome sequencing, as well as even reanalysis has a dedicated CPT code. Is the reimbursement sufficient? I can't comment on that, but definitely progress in the right direction.
Thank you so much. I know we're at time. It is 12:02, so lunch is up next, but I'd like to give a final thanks to Dr. Reddy and Dr. Kind.